here for chapter 13 for microbiology. And today we're talking about the microbe-human interactions. Very interesting chapter. Okay, let's go ahead and get through it. Okay, scoping out the chapter. First, we'll talk about, we actually have a microbial menagerie, which means our bodies are home to a complex community of tiny creatures that live and feed and reproduce there. All major groups are represented in the microbiota, from bacteria and viruses to fungi, protozoa, and archaea. This evolving coexistence has become an essential contributor to our nutrition, immunities, and general health. This is going to be an interesting slide. We'll get to it very shortly, but it shows the infection portals and pathways. Through the chain of infection, pathogens ultimately gain access to sterile tissues and grow there. Many exposed areas of the body provide an entrance that leads the way to vulnerable targets within the body. So this figure here depicts how the pneumococcus first enters the nasal passages and spreads from there to the lungs, ear, or brain. And also we're going to look at the tricks that invaders play on their host. So microbes come armed with a variety of special appendages, such as capsules, hooks, or fibers, for clinging to host cells and holding them in place. So then once pathogens are successfully established in the tissue, they can begin the process of invasion. Then we're going to talk about the toxins and enzymes and the damage that they actually cause. So once they reach a target site and begin to multiply, pathogens release factors that aid in their invasion or avoidance of host defenses. Many of these substances cause the damage that leads to signs and symptoms of disease. Then we look at how microbes travel between hosts. So in the later phase in the chain of infection, infectious agents are released from the body through exit portals, such as body fluids or excretions. This provides pathogens with a route to a new host and increases the chances for the microbe's survival and spread. So let's say this person has an infectious microbe and they spread it to another person. So their exit portal is through their mucus, and then they spread it, and through the mucus, it infects another person. That's an example of what they're talking about. Okay, keeping tabs on pathogens and disease. Infectious diseases can be monitored from many different perspectives. So epidemiologists aim to know their origins, how they are transmitted, who is infected, the cause of epidemics, the rate of infection, and the location of outbreaks and how they are recorded and reported. It's an epidemiologist. So let's start off with saying we are not alone. So the human body exists in a state of dynamic equilibrium. So many interactions between human body and microorganisms involve the development of biofilms. So colonization of the body involves a constant give and take. Here are some of the most significant interactions. So microbes provide a protection and stabilization effect on body surfaces. Microbes are involved in maturation of host defenses and development of the immune system. Microbes can invade and grow in sterile tissue, causing diseases. So let's talk about contact, colonization, infection, and disease. So microbes that engage in mutual or commensal associations with humans belong to the normal resident microbiota. Other terms you can use are indigenous, microflora, normal flora, and commensals. So the normal microbiota, normal flora, are the microorganisms that normally live on our bodies. And then a definition of infection. Infection is a condition in which pathogenic microbes penetrate host defenses, enter tissues, and multiply. Then, definition of a pathogen. is a microbe acting as an infectious agent. So a microbe that's a normal flora can actually be a pathogen if it gets into an area that normally does not have microorganisms. Then you have infectious disease. That's an infection that causes damage or disruption to tissues and organs. Know these definitions. And here we talk about how a microorganism can get exposed to and infect the body. So you have contact, then you have colonization. It starts to grow once it gets in. Then invasion, it crosses lines of defense and enters sterile tissues. Then you have infection. So that's where the microorganism multiplies in the tissue. So you can either have a carrier state where you don't have any signs or symptoms of disease, it's not apparent, but you're still infected, which means that you can actually spread to others. And then you see with this, you have the effects of the microbes result in injury or disruption of tissue, and then morbidity, mortality occur. Or the defenses can hold the pathogen in check, and then immunity or repair of the damage. So some of the resident microbiota. So it's the human as a habitat. So this is where a lot of microorganisms, a whole lot of microorganisms actually live. So internal organs, tissues, and fluids are microbe-free. So inside our body is free of microorganisms. So most areas of the body in contact with the outside environment, they harbor resident microbes. So, and we'll talk about what it means in terms of outside environment of our body. So you have transient microbes. Those are microbes that occupy the body for only short periods. Then you have resident microbes. These are microbes that become established. This is where they live. So here are some of the sites that harbor normal resident microbes. The skin and its contiguous mucous membranes. So you have the skin and any area that it flows with that goes not directly on the outside of the body, but let's say the mouth, now the lips, and it's continuous with the mucous membrane that goes inside the mouth. Same thing, when you have the urethra or the anus, the skin is on the outside and it becomes continuous with the mucous membrane as it delves further. And so upper respiratory tract, so the oral cavity, the pharynx, nasal mucosa, they harbor normal resident microbes. Gastrointestinal tract, so that's the mouth. And then the colon, rectum, anus. Then the outer opening of the urethra. Then you have the external genitalia. You have the vagina. And then you have external ear and ear canal. Then you have external eye, like your lids, lash, and follicles. So the origins of the resident microbiota. So generally stable, but it fluctuates with general health, your age, your diet, your hygiene, hormones, and drug therapy will change your microorganisms that live on your body. So in many cases, the microbiota benefits hosts by preventing overgrowth of harmful microorganisms. So that's called microbial antagonism. So endogenous infections occur when normal flora is introduced to a site that was previously sterile. So it's important to know that the microorganisms that live on our bodies will actually keep bad microorganisms from invading our bodies. But if a resident microorganism gets into an area that should not have microorganisms, then they can cause a lot of damage. That's where you have endogenous infections. So some of the sterile or microbe-free anatomical sites include the heart and circulatory system, the liver, the kidneys and bladder, the lungs, brain and spinal cord, the muscles, bones, ovaries, testes, glands like pancreas, salivary glands, sinuses, middle and inner ear, internal eye. Then the fluids within an organ or tissue are free of microorganisms as well. So the fluids in your blood, your urine, your cerebrospinal fluid, saliva prior to entering the oral cavity. So the salivary glands produce saliva and it's microbe free until it gets into the mouth where the microorganisms are in the oral cavity. And then the semen prior to entering the urethra. Now let's talk about the initial colonization of a fetus newborn, of the fetus and newborn. So uterus and contents are normally sterile until just before birth. So breaking of the fetal membrane exposes the infant. So subsequent handling and feeding of a newborn continue to introduce what will be their normal flora. So nature of the microbiota initially colonizing large intestine is influenced by whether the baby receives breast milk or formula. So we look inside the uterus, the baby is sterile, in sterile environment. And then as it passes through the birth canal, then microorganisms start colonizing the baby. If you feed the baby with a bottle, then it has microorganisms that go into the baby as well. If the baby breastfeeds, then microorganisms on the mother's skin, on her breast, and actually go into the baby. As other people handle the baby, the microorganisms that's on this person will get on the baby. And then even doctor's visits, as doctors handling and visiting with examining the baby, microorganisms are going into the baby. Now, all these um, typically are becoming his resident microbiota. These are the resident microorganisms. These are the microorganisms that he needs that will go to his gut, get on his skin, and become resident for this baby. Here's some of the indigenous flora. These are some of the microorganisms that actually are established and are in uh, different parts of the body. In the skin, and you look at um, the microorganisms that live there, and you have bacteria, fungi, arthropods, and then GI tract and the oral cavity, some of the things that actually live there. Large intestine and rectum, the type of organisms that live there. Upper respiratory tract, genital tract, urinary tract, the eye, the ear. I'm not going to ask specific names and where they're located for chapter 13. Here are some of the colonizers of the human skin and describing them. Skin is the largest and most accessible organ for microorganisms. So you have two cutaneous, which means on top of the skin, populations. First, you have transient. Transient means they just around for a little while, but then they're just there briefly. So they cling to the surface but do not grow there. They're influenced by hygiene. So the resident microorganisms are stable, they're predictable, less influenced by hygiene, and primarily are bacteria and yeasts. It just shows in terms of colonizer human skin, basically stay at the epidermis layer. And this is where you have a lot of dead skin. Does not get down into the dermal layer. This is sterile. This is an electron micrograph of some microorganisms that are uh, living among the dead skin. Now, the microbial residence of the gastrointestinal tract. The GI tract is a long, hollow tube bounded by mucous membrane, and tube is exposed to the environment. Uh, the GI tract is a long, hollow tube and is bounded by mucous membranes. So the tube is exposed to the environment. So variations in floral distribution due to shifting conditions like pH, oxygen tension, the anatomy. So you have oral cavity, you have the large intestine, and rectum harbor appreciable flora in the GI tract. So when we say that tube is exposed to the environment, it's a long hollow tube that goes from your oral cavity or your mouth all the way through your body till it gets to your rectum and anus, to the outside world again. Everything that goes through that long hollow tube is considered outside of your body. So that's what it means. The tube is exposed to the environment. So the inside of that tube is actually considered outside of your body. And this where it says the long hollow tube starts at your oral cavity, goes down to the pharynx, the esophagus, the stomach, duodenum, small intestine, and goes to the large intestine. And the large intestine comes and goes to the rectum and anus and out again. All inside of here is considered outside of your body. And if you notice the yellow portions, they're the portions where you have resident microbiota. So you have bacteria that will go in these different areas. And so in the oral cavity, in the pharynx, typically not down into the esophagus, and then also not in the small intestine, not in the stomach, but then you have the large intestine that has microorganisms, and then out to the outside world. So then the microbiota of the mouth. So this is the most diverse and unique flora of the body. So numerous adaptive niches and microhabitats. So you have different types of microorganisms that are in the cheek epithelium, the gingiva, which are your gums, your tongue, floor of the mouth, and tooth enamel. Each of them have their own type of microorganisms. The most common residents are aerobic Streptococcus species. And then teeth eruption establishes anaerobic habitat in the gingival crevice, favoring colonization by anaerobic bacteria. So there's where you get the dental caries and periodontal infections. So you can have bacterial count of saliva is 5 to 10 to the 9 cells per milliliter. A lot of bacterial cells. Now here's the microbiota of the large intestine. It has complex and profound interactions with the host being you. You have 10 to the 8th to 10 to the 11th microbes per gram of feces. That represents about 30% of the fecal dry volume. So 30% of the feces, if you don't have any water associated with it, 30% is actually bacteria or microorganisms. The intestinal environment favors anaerobic bacteria. So you have some of the ones that are anaerobic. So many species provide useful byproducts to hosts. So we have fermentation of waste materials in the feces generates different vitamins. Key in those are vitamin B12 and vitamin K. Then you have bacterial digestive enzymes that convert disaccharides to monosaccharides, or they promote steroid metabolism. So intestinal bacteria contribute to the intestinal odor. Now, the inhabitants of the respiratory tract. You have in the respiratory tract, you have oral streptococci. These are the first organisms to colonize. So you have in the nasal entrance, the nasal vestibule, and the anterior nasal pharynx. And you can have Staphylococcus aureus that is in that area. And then mucous membranes of the nasal pharynx are like the Neisseria species. The tonsils and lower pharynx are shorted streptococci and Haemophilus. So then you don't have any flora in the bronchi and lungs. So here's where, with the respiratory, it has in the nasal vestibule, nasal cavity, the pharynx, and it goes down to a portion of the trachea. These are the areas that can have normal microorganisms. And as you see, as it gets down to getting into the lungs, you do not have microorganisms that venture that far. So now we have the microbiota of the genital urinary tract. So sites that harbor microflora are in females. You have vagina and the outer opening of the urethra. With males, you have the anterior urethra. So the internal reproductive organs are kept sterile through physical barriers, like with the cervical plug for females. So then you have the kidney, ureter, the bladder, and the upper urethra, kept sterile by urine flow and by the bladder emptying. So changes in physiology influence composition of normal flora. So for example, in the vagina, the estrogen, the glycogen, the pH of the vagina kind of control how many and how much of the microbiota are thriving. So here's the female. This is the urethra. And you see at the urethra, here's the urinary bladder. And as you void, it clears any microorganisms that can get up a little bit further. But for the most part, at the outer opening of the urethra, then you can have microorganisms. And then you have a larger opening, which is the vagina and the vaginal vault, and microorganisms normally inhabit this area because it's directly associated with the outside world. So you have resident microorganisms that are in this area, but it doesn't get to the place where it goes into the uterus because of physical barriers, like you have uh, plugging up the cervix to not get into the uterus. With the males, this is on the outside. You have microorganisms on the outside, and when it goes to the anterior portion of the urethra, an anterior portion of the urethra, you'll have some microorganisms right at the tip. But as you go further up, you don't have any. And this urethra is cleared by the urine that flows and pushes microorganisms out. So a maintenance of normal microbiota. Normal flora is essential to the health of humans. So the flora creates an environment that may prevent infections and can enhance host defenses. So antibiotics, dietary changes, and disease may alter the flora. Probiotics, probiotics introduce known microbes back into the body and tries to get the microorganisms back on track. So here's a concept check. So normal resident microbes are absent from the what? Are they in the pharynx? Yeah. Are they in the lungs? No. Are they in the intestines? Yeah. Hair follicles? Yes. Mouth? Yes. So the answer would be they're absent from the lungs. So major factors in development of infection. So finding a portal of entry is very important. And so the portal of entry for microorganisms to get in is skin, GI tract, respiratory tract, urogenital tract, and endogenous biota. So these are uh, microorganisms that are already on your body and known to be on your body, but for some reason found a way into your body. Then they attach firmly. So they attach using fimbriae, capsules, surface proteins, viral spikes, different hooks. Then once they get in, they have to survive the host defenses. So one of the major functions of our defenses, our immune system, is phagocytosis. That's where we engulf different microorganisms. So one of the defenses that microorganisms have, they try to avoid phagocytosis. Then another one, some microorganisms, they allow themselves to get phagocytized or engulfed by a um, immune response, but they avoid death inside the phagocyte. Some microorganisms have been able to do that. Then others just try to evade the actions of the immune system completely. So here are some of the causes of damage and disease. First, you can have direct damage. The microorganism can release toxins, enzymes, or actually cause cells to burst. Then you can have indirect damage, where the host response is inappropriate or excessive. So we can have a problem where you have even multi-organ failure because your body overcompensates and excessively tries to fight off a microorganism inappropriately and will cause damage to your own cells with your own immune system. Then another factor in development of infection is the microorganism has to find a way out of your body so it can infect someone else. So you have portals of exit. So you have the respiratory tract, salivary glands. That's where you have transmitting through mucous membranes, where you actually, mucus leaves one person, and that's a portal of exit for a microorganism through the saliva. Or you can have portal of exit is through the skin cells. Infected skin cells get on someone else and spread. It can be spread through fecal matter. Diarrhea can be full of microorganisms, and if that gets in someone else's mouth or in someone else, then they could actually spread the microorganism. Urogenital tract, that's where you have a lot of sexually transmitted diseases. It goes from one person to another, and the microorganism would be in the uh, fluids or on the skin of one person and gets onto another. Then, of course, blood. It's in the bloodstream of one person and somehow gets transmitted to another person. Okay, here's some major factors in development of infection. So, factors that weaken host defenses and increase susceptibility to infection. So, old age and extreme youth. So, infancy, prematurity with extreme youth, and old age. As you get older, your immune system gets weaker. Then you have genetic defects in immunity and acquired defects in immunity. So people that are immunocompromised, they have genetic defects or they have defective immune system. They're at risk where the host defenses are weakened. And so you're more susceptible to infection. More susceptible when you have surgery or organ transplants. Organic disease like cancer, liver malfunction, diabetes, you're susceptible to have immunocompromised. If you're in chemotherapy, anything that suppresses uh, your immune system, immunosuppressive drugs. If you're under physical or mental stress, that can suppress your immune system. And other infections. So if you have one infection, another one has more of a tendency to be able to attack. Okay, then you have what we call true pathogens. True pathogens are capable of causing disease in healthy persons with normal defenses. So, influenza viruses, the plague bacillus, malaria protozoan, you get exposed to it, they are capable of causing disease in even healthy persons. And then you have opportunistic pathogens. These are pathogens that normally would not cause disease, but they cause it if the host defenses are compromised or they grow in part of the body that is not natural to them. So this is opportunistic pathogens like Pseudomonas and Candida albicans. Severity of disease depends on the virulence of the pathogen. So how severe it is depends on the virulence. So virulence factor is the characteristic or structure that contributes to the ability of a microbe to cause disease. Virulence is characteristic or structure that contributes to the ability of a microbe to cause disease. That's the virulence factor. Here's some of the portals of entry. Portals of entry are the characteristic route a microbe follows to enter the tissue of the body. So you can have exogenous agents. So that's exo usually means outside of. So they originate from a source outside of the body. And so endogenous agents already exist on or in the body. This is your normal flora. So if they're already on or in your body, they're considered endogenous agents. Exogenous agents are ones that are not part of your normal flora. And so This just shows some of the areas that you can actually get diseases becoming established. So you always have, these are different areas that have normal microbiota on the skin, the respiratory tract, gastrointestinal tract, or the conjunctiva of the eye, in the urogenital tract. Uh, and so the gastrointestinal tract goes all the way down and out to the radius. So those are normal areas. So here's some of the portals of entry. With the skin, if you get nicks or abrasions, punctures, or incisions that can break the barrier. Gastrointestinal tract, you can get it through eating contaminated food, drink, and other ingested material that have microorganisms. Respiratory tract, through the oral nasal cavities. That's like this coronavirus that we're dealing with currently. This is 2020. Is uh, you get exposed to you through mucous membranes in the oral, your mouth, and your nose, and actually your eye as well. Any mucous membrane. And then you can get it through your genital tract. So sexual contact, displaced organisms that should not be there can actually cause it. Transplacental crosses through the placenta, gets to the baby. This is data. I'm not going to ask you this, but you can actually look through this. It just shows estimated number of new cases per year, estimated number of cumulative cases over extended time frame. And so you can look at some of the numbers with that and some of the common things. Okay, so here's some of the pathogens that infect during pregnancy. And actually, they use the acronym STORCH, stands for syphilis, toxoplasmosis, other diseases like hepatitis B, AIDS, and chlamydia, rubella, cytomegalovirus, and herpes simplex virus. These are things that can infect the unborn child through the placenta. So requirement for an infectious dose. So what is an infectious dose? Infectious dose is the minimum number of microbes that are required for infection to proceed. So the minimum number of microbes required for an infection to proceed. So microbes with small infectious doses have greater virulence. They don't need as much of it to actually cause an infection. So lack of infectious dose will not result in infection. So if you don't have the minimum number of microbes required for an infection, then you will not get infection even when you're exposed to a microorganism. Okay, this just shows the estimated infectious dose of selected pathogens. You look measles, if you just get infected or you have one virus, then you could actually have catch the measles. But you look at some of these, you look at anthrax, you need 8,000 to 50,000 spores before you actually would catch anthrax. Okay. Okay. Now attaching to the host. So adhesion. So the microbes gain a stable foothold at the portal of entry wherever they get in, dependent on binding between specific molecules on host and pathogen. So some microorganisms have fimbriae, or flagella, glycocalyx, cilia, hookers, hooks, and barbs that they utilize to actually grab onto the host. And here's some of the common ones that how they actually grab onto different parts of your body. Like HIV causes AIDS, and the adhesion has viral spikes adhere to white blood cell receptors. And so when we talk about this SARS-CoV-2, which we're dealing with, which is if this was up to date, they'd actually have the microbe would be SARS-CoV-2, and the disease it caused would be COVID-19, and the adhesion mechanism it would be viral spikes adhere to ACE2 receptors on alveoli. Okay. All right. So now, now that they've attached and they invading and become established, and a lot of microorganisms have exoenzymes. This is where bacteria produce extracellular enzymes that dissolve barriers and penetrate through or between cells to invade underlying tissues. So these are the microorganisms, and they release all these particles, which are enzymes that will eat through this protective cell cement. And then they'll be able to break through and get to the underlying sterile environment. Then some of them release toxins. So toxins, and they're primarily exotoxins, secreted by bacteria, damage the target cells, which die and begin to slough off. Now these bacteria secrete exotoxins. Once these cells die off, now the bacteria can get to the underlying sterile environment. Then some of them, in terms of getting established, some of them are able to block phagocytic response. Like we have macrophages, and part of our immune system, that's one of some of our front line that will attack and engulf invaders. Well, some bacteria, what they do is they actually have a property that enables them to escape phagocytosis and continue to grow and cause further infections. See, this is how it should work, where the phagocyte will engulf a bacteria. But then some bacteria have a capsule that do not allow the phagocyte to engulf them. So with this capsule over it, the phagocyte cannot engulf it. The bacteria will be free to move around and invade tissues. And here's some of the invasion factors. Some bacteria have these fimbriae that attach to microvilli, and then when they attach to the microvilli, they're able to secure themselves, inject, and release proteins into the cell. And then when they inject into the cell engulfs the bacteria. Now, when the cell is pulled into a vacuole, now it multiplies internally and then moves out of the cell and into deeper tissue. And that's where you'll get invasion into sterile areas. That's how some of them get through the cell. So here's some of the invasion factors. Salmonella has a unique system to invade intestinal cell. So after adhering to the cell's microvilli, it's just describing what we just went through. It injects protein in the cell with a probe-like structure. These proteins disrupt the actin filaments and cause the cell membrane to form a series of ruffles that surround this bacterium and pull it inside. It becomes shielded within a vacuole and begins to grow. So from here, it can gain entrance to nearby tissues. Just describe what we just saw. Now you have bacterial toxins. Toxin is a specific chemical product of microbes, plants, and some animals that has poisonous effects on other organs. Toxinosis, that's adverse effects of toxins. So you can have toxemias, so that when the toxin is spread by the blood from the site of infection. So you have a toxemia where the uh, the toxin is secreted at one area and then it's spread in the blood and affects everything. That's what you get with tetanus and diphtheria. And you have intoxications that's caused by ingestion of toxins. And that's like with botulism. You'll actually ingest it as opposed to it running through your bloodstream. So types of bacterial toxins. You have the endotoxin, which is not secreted, but it's released after a whole cell is damaged. So with an endotoxin, it's composed of part of the outer membrane of a gram-negative cell wall. So it's not secreted, but after the whole cell is damaged, the endotoxin is released from the bacteria cell wall. exotoxins secreted by a living bacterial cell into the infected tissue. So it has strong specificity for a target cell, disrupting its membrane. So this is where the bacterial cell secretes the toxin directly. This is after the bacterial cell is dies, then the endotoxin affects the host cells that are left around it. And they talk about the exotoxins and endotoxins and the effects on different parts of the body. So target organs are damaged, like heart, muscle, blood cells, intestinal tract. They show dysfunctions. Uh, and so general physiological effects, like you can get fever, malaise. This is where the endotoxin, this is the bacterial cell, once it's dead, then this cell actually releases the toxins into your body. And it compares the exotoxins and the endotoxins. And so you can look through it, but I'm not going to ask you specific questions on these charts. So actions of AB toxins. AB toxins consist of two parts, an A, which is active component, and a B, which is a binding component. So the B component binds to a specific receptor molecule on the surface of the host cell. Then the A component is moved across the host cell membrane. And we'll describe that in the next slide. The A component inhibits the cellular protein to cause damage. So this is what it's talking about. This is the A part, and this is the B part. So what happens, there's a receptor, and the B part attaches to the receptor. So once the B part attaches to the receptor, then it's able to gain access. And so now this comes in, the A and the B come in, but then the B stays where it's attached to this engulfed portion of the cell membrane, but the A part is released. Now, the toxin that was the A part acts on ribosomes and blocks protein synthesis, causing this cell to die. So there's an A part and a B part. The B part just gains access. The A part is the one that actually causes the disruption. Okay. And so only gram-positive bacteria can secrete exotoxins, true or false? That is false. Remember, it's the gram-negative cell wall, our cell membrane. Okay, so now we're talking about the process of infection and disease. So 4 distinct stages of clinical infections. First, you can have incubation period. That's the time from initial contact with the infectious agent to the appearance of the first symptoms. So the agent is multiplying, but damage is insufficient to cause symptoms. So you can have several hours, several years before you um, show results of infection. That's incubation. So during that time, they can actually spread the disease because it's in there, but they don't show any signs or symptoms. Then you have what we call a prodromal stage. So you have vague feelings of discomfort, non-specific complaints. That's a prodromal stage. Then you have what they call a period of invasion. So the organism multiplies at high levels, becomes well established, more specific signs and symptoms. So you don't have the vague signs and symptoms, you actually full fledged are getting sick, more specific signs and symptoms. Then you have the convalescent period. So as the person begins to respond to the infection, and so your body's fighting it off and you feel better, and the symptoms start to decline. All of these periods, you can transmit the infection. So from the time of incubation, you don't even have any signs or symptoms. Then when you're vague feelings, you're infectious. Then of course you know when you're full-fledged sick. But then when you start to get better, you can still spread infection. Okay, this just shows in terms of initial exposure, you don't have any, so the intensity of the symptoms as it goes up. So you don't have any symptoms, incubation period. Then you start feeling vague symptoms, just a little bit. And then you start really feeling bad. and it gets up to a height where you feel really bad. That's the height of infection. All this is period of invasion. Then you feel really bad, and you stay feeling really bad for a minute. And then your body starts fighting it off. You start getting treatment. Then you have convalescent period until you feel back to normal. But all this time that even when you're feeling better, getting back to normal, you can still be infectious. Because so the patterns of infection, you can have localized infection. So the microbes enter the body and remain confined to a specific tissue. That's localized infection. Then you can get systemic infection. The infection spreads to several sites and tissue fluids, usually in the bloodstream or through the bloodstream. That's where you get systemic. Then you get focal infection. That's where the infectious agent breaks loose from a local infection and is carried to other tissues. And we'll get a chart that kind of shows these. Then you can have mixed infections. So several microbes grow simultaneously at the infection site. So you get a compromise and a site where you get an infection, and if there's multiple microbes that are available, multiple microbes will actually attack and infect. That's what you call a polymicrobial infection. That's mixed infection. So according to the sequence of occurrence, that's how you can, the pattern of infection can be identified according to the sequence of occurrence. So primary infection is the initial infection. This is what first started it all. Secondary infection is subsequent infection by a different microbe. So one microorganisms cause a certain sickness, and then because your immune system is compromised, or because that one caused a certain amount of damage, a second one was able to attack and attach. That's primary and secondary infection. Then you have what we call acute infection. So acute infection comes on rapidly and severe, but it has short-lived effects typically. Then you have chronic infections. So it progresses and persists over a long period of time. Here's some of the patterns of infections. Let's say you get a boil or a cut and it gets infected, it's localized. Systemic infection, it gets into the bloodstream and goes everywhere in your body. So this is a focal infection. You can have an infection in your throat, let's say, and then it goes and affects a different organ. Okay? Then you can have mixed infection. If you get a disruption in your gastrointestinal area, there's various microbes, and so multiple microbes will infect at that area. And then this is where you have a secondary infection. This, like if you have a bacterial infection in the urinary bladder, and then you may get a secondary or a yeast infection in the vagina because you treated the bacteria in the bladder, and it kills all the bacteria. Now the bacteria is dead in the vagina that are beneficial, and the yeast overrun the vagina because it does not have the bacteria to hold it in check. So know the difference between a sign and a symptom. So with signs, it's objective evidence of disease as noted by an observer. So a sign is something else someone can see and identify that you have an issue. Symptom is more subjective. Subjective evidence of disease as sensed by the patient. So here's some of the common signs and symptoms of infectious disease. So a sign, something can take your temperature and you have a fever. Oh, we can tell that your symptom is I feel cold. So you get chills. So you see the different things that are signs and symptoms. Signs, things that we can monitor and see. These are things that you feel. Yeah. So the earliest symptoms of disease as a result of the activation of the body defenses. So these are the result of the body's defenses. These are symptoms. These are what you feel in response to your body fighting something off. So the fever, the pain, the soreness, the swelling. And so some of the signs of infection are the um, edema, So accumulation of fluid, and you can get the swelling with that as well. And you'll see a granuloma or abscess, so walled off collection of inflammatory cells and microbes. Sometimes it gets so big, invading microorganisms, that our body walls it off from the rest of the body to make sure it doesn't get any further. And so you have, so those are some of the signs. Lymph and Lymphitis, that's a swollen lymph nodes. That's a sign of infection. And some of the signs in the blood. So it changes in the number of circulating white blood cells. So you can have leukocytosis, which is leukocytosis an increase in white blood cells, saying that the body's trying to fight something off. Leukopenia is a decrease in white blood cells. Certain diseases will actually have a decrease in white blood cells. Septicemia, if you see microorganisms that are multiplying in the blood and present in large numbers. So that's septicemia. Now, you can have bacteremia, so that's a small number of bacteria that are present in the blood, but they're not necessarily multiplying. That's bacteremia. Okay? And that's a special type of septicemia. Viremia, small number of viruses that are present, not necessarily multiplying. So you'll have viremia. But when you have septicemia, when you're septic, you actually, microorganisms are multiplying in the blood. They're present in large numbers. So sometimes you have infections that go unnoticed. It is rather common for infection to produce no noticeable or typical symptoms. So that's called asymptomatic or subclinical infections. So even though someone's infected, the host doesn't show any signs of disease. So inapparent infection, so the patient experiences no symptoms, and so they don't seek medical attention. Now let's look at some of the portals of exit. We looked at the portals of entry, and we talked about infection. Now let's look at portals of exit, because pathogens depart by a specific avenue. So that influences the dissemination of infection. This will affect how much it will affect other people, how much it will spread. So it has to have a way out of one host so it can get to another host. So you have respiratory and salivary portals. This is the mucus, sputum, nasal drainage, saliva. These are all portals for it to get out of one host and into another. Epithelial cells. This is on your skin and scalp. And so let's say someone has leprosy, and the microorganism that causes leprosy. Well, the skin can transmit, has the leprosy on your skin of one person, and they touch another person, and it can spread to the other person. You can have fecal exit. That's through diarrhea, feces. That's the microorganisms are in the diarrhea or in the feces, and if someone else touches it, somehow gets that into their mouth, then the microorganism can spread. Urogenital, that's through the urine, through sex. That's typically the um, sexually transmitted diseases are through urogenital tract. Then removal of blood or bleeding. So someone that has a disease in their blood, and they get their blood onto someone else, and that person is susceptible or has an opening that gets into their blood. And so here's some of the a figure that shows some of the portals of exit: coughing or sneezing, an insect bite, that's through the blood. So it takes some of the blood from one person and goes and infects another person. Skin cells, you have an open lesion, uh, and in the urine, through the feces, through removal of blood. These are portals of exit. So now you look at the persistence of microbes and pathologic conditions. So apparent recovery of host does not always mean that the microbe has been removed. Think about that. Apparent recovery of the host does not always mean that the microbe has been removed. So sometimes you can get latency. So after initial symptoms in certain chronic diseases, the microbe can periodically become active and produce a recurrent disease. So a person may or may not shed it during that latent stage. So the latency, that's like with chickenpox. Chickenpox is varicella zoster. So you get better from chickenpox. You have immunity, lifelong immunity, typically from chickenpox, but the virus, the varicella zoster virus, is latent. It actually incorporates its genetic code into parts of your genetic code in certain cells of your body, and it just keeps just sitting there replicating. It sticks its head out every now and then. Your body remembers it and breaks it down, and you don't have a problem with it unless it gets to a place where your immune system gets compromised, like you get sick with something else, or you get older, your immune system is decreased. Then, when it sticks its head out, the immune system isn't able to fight it off as effectively, and you will have an outbreak of shingles, which is caused by the same virus that causes the chickenpox, varicella zoster. But that's an example of latency. Syphilis has latent periods. And so it seems like you get better, but then when it comes back with more greater force. Then you have chronic carriers. This is a person with a latent infection who sheds the infectious disease. Latent infection means that the disease is not causing signs or symptoms, but they're still infectious. They're still spreading it. You should look up typhoid Mary. Typhoid Mary was a great example of spreading disease, and she did not show any signs or symptoms of it. Then you have what they call sequela, long term or permanent damage to tissues or organs. So, epidemiology is the study of the frequency and distribution of disease and other health related factors in defined human populations. It involves many disciplines, not only microbiology, but also anatomy, physiology, immunology, medicine, psychology, sociology, ecology, and statistics. It considers many diseases other than the infectious ones. So it looks at heart disease, cancer, drug addiction, and even mental illness. Now, the origins, and the transmission patterns of microbes. So, if you call something a reservoir, that's a primary habitat of pathogen in the natural world, where it actually is and where it is stored. So, human or animal carrier could be in the soil, could be in water, plants. And then the source is the individual or object from which an infection is actually acquired. So a lot of diseases have reservoir. They are in certain areas, and that's where they kind of grow. And they don't necessarily cause an infection because they're not exposed to, uh, the different hosts that they're waiting for. But they sit and they wait, and they're in reservoirs. Then you'll have the source is the individual object from which an infection is actually acquired. Okay. So you can have living reservoirs. One could be a carrier, individual who inconspicuously shelters a pathogen and spreads it to others. That's where we're talking about typhoid Mary. She was a carrier. So you can have asymptomatic carriers, which means that they spread it and they don't show any symptoms whatsoever. Then you can have a passive carrier. So a contaminated healthcare provider transfer them to other patients. And we'll show you a picture of that. There's a picture of all these. This is asymptomatic. So they have a microorganism in their body and they go about their daily life and they spread it without even knowing they're spreading it. And this is a passive carrier. This is like this person's sick, has a microorganism, and let's say there's something in the feces or urogenital. This healthcare worker helps them, but they get exposed to this microorganism. As they get exposed, they come to another patient and they give that microorganism to another patient who's immunocompromised. And so this person did not get infected, did not get sick, but they transferred this microorganism and took it to another person. This is passive. Then you can have incubation carriers. So remember we talked about incubation? This is where someone has uh, a microorganism, but they really have limited uh, signs or symptoms. So the spread of infectious agents during the incubation period, they can still spread it. Convalescent carriers, so these are people that are feeling better, and so they feel better and they aren't actively sick, so they think, okay, I'm all right. I'll go back to work or I'll go to the gym. I'm feeling better, so I'll work out and it should help me sweat this thing on out. Well, they could actually still be spreading it. They're living reservoirs. They're better. Their immune system's fighting off effectively, but they still spread it to other people. Then you have the chronic carrier. That's an individual who shelters the infectious agent for a long period of time. And this is where they talk about Typhoid Mary, where she spread typhoid fever, which is very detrimental to a lot of people. She was a cook, and she would spread it to other people without her having any symptoms, but she spread it to other people. So if a nurse transfers a pathogen between patients without becoming infected herself, the nurse has acted as the, and which ones are these? A chronic carrier, a convalescent carrier, incubation carrier, asymptomatic carrier, passive carrier. And of course, it's passive carrier. And so know what each of these are, though, and how they actually look. Okay, then we talk about vectors. A vector is a live animal or other than a human that transmits an infectious agent from one host to another. So majority of vectors are arthropods. Arthropods are like fleas and mosquitoes, ticks, and flies. So some larger animals can also spread infection, like some mammals, birds, lower vertebrates. But majority of vectors are arthropods because they'll, let's say a tick will bite one animal that's infected, and then they will carry that to another individual. So you can have a vector, can be a biological vector, is actively participates in a pathogen's life cycle. So whatever that microorganism is, they actually, uh, when the vector picks them up, then that microorganism will actually increase or continue on with their life cycle, and they know they're about to be injected into something else to cause infection. So let's say they may be, the microorganism may be inactive, but then when it gets into the vector, then it will actually mature and know it's going to be ready to infect something else. That's a biological vector. Mechanical vector is not necessary to the life cycle of that microorganism or infectious agent and merely transport it without being infected. So that's my mechanical vector. Transports it without, it's not infected at all, it just transports it. Then we talk about zoonosis. So infection indigenous, which means they're normal in animals, uh, infection indigenous to animals, naturally transmissible to humans. Okay, so at least 150 zoonosis exists worldwide. So 70% of all new emerging diseases worldwide come from animals. So it's impossible to eradicate these diseases without eradicating the animal reservoir. So the animal holds on to it and transmits it to humans. That's where they call zoonosis. And it looks at animal reservoirs and their sources. So these are common zoonotic infections: rabies, yellow fever, viral fevers, all the influenzas, the Rocky Mountain spotted fever. These are things that we get from animals. And it just continues on with more of them. I'm not going to ask you specifics for these. Okay, now Let's look at what it means for communicable disease. When an infected host can transmit the infectious agent to another host and establish an infection in that host, that's called communicable disease. Non communicable disease means that infectious disease does not arise through transmission from a host to host. So that occurs primarily when a compromised person is invaded by his or her own normal microflora. So if a person that normally is not sick, but it gets sick because of the normal uh, invasion of their body by their normal microorganisms that are on their body. You can also get a non-communicable disease when contact with the organism in natural non-living reservoir. So you run across something that is in its normal area, like say in the environment, and that's how you catch it. You actually catch it from directly from its natural reservoir. You don't catch it from someone else. You get it from the environment. So you can have patterns of transmission. You can have direct contact, so you have physical contact or fine aerosol direct droplets, or you can have indirect contact, so that it passes from infected host to an intermediate conveyor and then to another host. So what you can do with that, if someone's infected, let's say if we look at this uh, coronavirus that we're dealing with right now, someone's infected, they wipe their nose, then they take the money out of their pocket, they hand their money to the cashier. So the cashier has the money, and so they get the virus on their hand from the money, and then they give the money to another person. Other person takes the money, and then as they're leaving out of the store or gas station or whatever, then they rub their nose. The virus that was on the money now went to their hand, and now they rub their nose, or they eat a candy bar and lick their fingers. Now that virus is in them. So that's indirect contact. Passes from infected host to an intermediate conveyor. In this case, the intermediate conveyor would be the money, and then to another host. So the vehicle is an inanimate material like food, water, biological products, fomites. Fomites would be the money. Fomites would be a handle on a door. Fomite would be, um, your steering wheel would be the keys on a keyboard. Someone uses the key on the keyboard, puts some virus on it, and then you come to the key on the keyboard and type in the keyboard, and then you touch your face without washing your hands first, and that vehicle was the keyboard, and that's called a fomite. Then you can also get it droplet airborne, droplet nuclei aerosol. Here's a picture of someone sneezing that is, they didn't try to cover, they didn't try to stop it. And this is the sneeze and how the droplets, see how the droplets are going down fast? And the droplets with this coronavirus, they has to be on mucus. And so all this is mucus. And so the coronavirus could be on this one if this person was infected with coronavirus. These droplets pretty much stay airborne for about 4 feet, and this can spread about 4 feet. And then the droplets drop onto surfaces, onto the floor. We have not identified that coronavirus, the SARS-CoV-2, remains airborne for any length of time. These droplets, the coronavirus, to be infectious, the infectious dose has to be on mucus, and this mucus will fall after about 4 feet. That's why we have social distancing of 6 feet. So, how communicable infectious diseases are acquired? So, communicable infectious disease, you have direct transmission, directly, directly, vertical transmission from the mother to the baby, biological vectors. This is direct transmission. Then you have indirect transmission. This is fomites. These are contaminated inanimate objects. You have the uh, the person who's in the food, indirect transmission is this person is infected, touches your food, then you eat the food. And so then you'll have indirect transmission. Then you'll get droplets, a nuclei, the dry airborne respiratory secretions. That's like you're breathing in. This is now tuberculosis is airborne. And if you breathe in airborne particulates, and then you'll actually get infected. And then airborne microbes, like from different birds and rat feces, then it becomes aerosolized, and you can breathe that in and get sick as well. Those are all indirect. Okay, work of epidemiologist. And so you can look in terms of surveillance, reporting diseases, talks about the CDC and the World Health Organization. I'm not going to ask you questions regarding epidemiologists, but it's interesting to look and follow up on that. And it talks about the frequency of cases, prevalence, total number of existing cases with respect to the entire population, usually represented by a percentage of the population, how prevalent it is. Incidence is measures the number of new cases over a certain time period. Mortality rate, that's total number of deaths. When you talk about mortality rate, total number of deaths. Morbidity rate, total number of people afflicted with a certain disease. So you look at mortality and morbidity rates, how many people are affected and how many people were actually died from it. And then it talks about this talks about pertussis specifically, which is pertussis whooping cough. And then talk about Ebola. And you look at the numbers in terms of the cases that were identified. And then you look at the epidemiologist, looks at the different cases of this particular situation, is looking at the HIV infection. Like I said, I'm not going to ask you about much about epidemiology, but these are some of the patterns of infectious disease occurrence. You have endemic occurrence. That's disease that exhibits a relatively steady frequency over a long period of time in a particular geographic location. That's endemic. Sporadic, occasional cases are reported at irregular intervals. So this is endemic. This is certain areas and it has number of cases, but it's in this area. And each one of these circles represents an outbreak. So you have outbreak of a certain disease in this area. That's endemic. Sporadic is where it just shows up sporadically. There's no given area where it shows up consistently. It's sporadic occurrence. Then you have epidemic. That's when prevalence of a disease is increasing beyond what's expected. That's epidemic. Pandemic, epidemic across continents. So it in prevalence of disease, it increases beyond what you're expecting. But then when it's beyond what you're expecting across continents, then it's pandemic. It's across the world. So in this is like if you have flu, and you have flu here, but then you have flu, and you have flu all over the nation. Pretty much flu, that's an epidemic. So you can have a disease that is spread out, not in one general area, but everywhere in your country. That's an epidemic in your country. Now, if you have cases here, you have cases here, you have cases here, you have cases here across continents, that's a pandemic. This is where you start seeing cases in different countries. Pandemic. Okay, now you can have an outbreak, which is a small scale epidemic in a more limited area. So you can have a point source epidemic, so the infectious agent from a single source. Then you can have a common source epidemic, all cases exposure to cases from exposure to the same source. Then you have propagated epidemic that shows sustained increase over time indicating that it's being communicated from person to person. And you can see these. This here shows the point source epidemic. It comes from one person, and that's the only place that it actually affected a certain number, certain amount, and that's all. This is a common source, so one person is affecting a lot of people, and so but it's all coming from the common source, and not one person. One event, let's say, is coming causing epidemic from one source. And then this is propagated epidemic. This is it starts here, but then it goes and it starts going, and it has a trajectory that is exponential. It means that it's being transmitted from person to person. So then you have nosocomial infections. These are also called healthcare associated infections. Nosocomial infections are healthcare associated infections. They're diseases that are acquired or developed during a hospital stay. So you get them from surgical procedures, from equipment, from personnel, exposure to drug resistant microorganisms, anytime that you get an infection from a hospital. The most common involves surgical incisions and the respiratory tract, GI tract, skin, urinary tract, and from blood. Then you have gram negative intestinal flora, gram positive bacteria, and then true pathogens such as the tubercle bacillus, salmonella, hepatitis B, influenza. You'll get those in nosocomial infections. So we have about 2 to 4 million cases per year in the United States. So they have approximately 90,000 deaths from hospital acquired infections. And this is just a chart that looks at the types of infections that you can get from the hospital. And the reason that a lot of hospital acquired infections are so prevalent is because when people go to the hospital, they're typically immunocompromised. And so if they get exposed to just about anything, they're susceptible to catch it. And so the prevention of nosocomial infections, you look at what we actually do. Type of isolation. So enteric precautions, respiratory precautions, drainage and secretion precautions. And these are some of the things, protective measures that are done. Gowns and gloves, private rooms, gowns and gloves for drainage, and they prevent the spread of these diseases. Then you can have strict isolation and reverse isolation. Same as strict isolation, but room may be ventilated by laminar airflow filtered through a HEPA filter that removes the airborne pathogens. So the infected persons must be barred from going out to other places, other people. Okay. Okay. Now you have universal precautions. These are stringent measures to prevent the spread of nosocomial infections or hospital-acquired infections from patient to patient, or also from patient to worker, and from worker to patient. Use universal precautions. So it's based on the assumption that all patient specimens could harbor infectious agents, so must be treated with the same degree of care. So if prevention is not possible, they include techniques to take purposeful measures to decontaminate potentially infectious material. This is how they define universal precautions. Act like whatever fluid, whatever situation you're in, if you're going to be exposed to certain fluids or certain things, assume that it has a bad microorganism associated with it and protect yourself. So, concept check. The occurrence of Lyme disease the occurrence of Lyme disease mainly in areas where certain species of ticks live would define it as a what type of disease? If the Lyme disease is only where certain species of ticks live. Very good. Endemic. It's endemic to that area. Very good. All right. So that's the end of the presentation. Just wait a couple minutes and I will go over the what I want you to kind of emphasize for this exam. Okay, let's go ahead and do a review for the quiz. Now, definitely know the definition of an infection. Let's see, know the different types of flora that we have, like normal flora, indigenous flora, pathogenic flora, um, the different types of flora. Know how uh, we get colonized with normal flora. Where does that occur? How does it occur? Know some of the um, benefits that normal flora, resident flora benefits our body, and where typically we benefit. Know the different stages of infectious disease, like prodromal and incubation and invasion. All those different stages. Um, Couple questions on that. Know definition of like inflammation, sequelae, toxemia, syndrome, latency period, what those definitions are. Know what the definition of epidemiology is, what an epidemiologist does. Know the different carriers, names of the carriers, and know how things are transmitted, like you know, fomites and vectors and reservoir, source, carrier, know the definition of those. A couple different questions on that. Know what vertical transmission is. Vertical transmission, that's when you transmit from the mother to the fetus. And know the animals that participate in the life cycles of different pathogens. And then know, like, once again, the definition of a vector, know the difference between mechanical vectors and biological vectors. And then know what hospital acquired infections are called and how they occur. Um, let's see, know how uh, diseases occur, whether they're epidemic, endemic, pandemic, sporadic. Know the different definitions of how diseases occur. And then know how and why a resident a resident resident microbe or resident flora, what it means when they actually cause disease, opportunistic infections, and the definition of septicemia and bacteremia, viremia, and no definition of what a pathogen actually is. Know about portals, portals of exit for microorganisms, and no definition of universal precautions and how it is that keeps infections at bay. Okay, we're going to talk next about uh, we're going to go through chapter 18, and that'll be very brief. Okay, this is chapter 18, which is the cocci of medical importance. And we're actually going to zip through to slide number 4. What I'm going to do is the quiz number 3, is going to be 20 questions from chapter 13. And then I'm going to have 5 extra credit questions. So really, it's going to be 5 extra questions, but if you don't get them, then it doesn't hurt you. You can still get however much out of 20 on this quiz from chapter 13. But if you take your time and memorize these slides 4 and 5, then you can get 5 extra credit points. Okay, so these are the cocci of medical importance. So we have Staphylococcus aureus, and so for the skin or skeletal system, diseases they cause are boils, carbuncles, impetigo, scalded skin syndrome, osteomyelitis. And if it gets into the cardiovascular lymphatic system, endocarditis, toxic shock syndrome, gastrointestinal food intoxication, and respiratory, you can get pneumonia, all from Staphylococcus aureus. Staphylococcus aureus is a very strong microbe, and it's very beneficial to us. It helps protect us from other microorganisms that could do a lot of harm. And so this is one of our resident microbes, but it it helps us when it stays outside of inside of our body. All these things occur when it goes into areas that it should not go in, where it should be sterile. And then we have coagulase negative staphylococci. You can get these in surgical infections. You can also get endocarditis. It can cause urinary tract infections. Then you have group A strep, which can cause impetigo, uh, erysipelas, necrotizing fasciitis, scarlet fever in the cardiovascular or lymphatic system. You can get rheumatic fever. If it's in the respiratory system, you can get strep throat, sinusitis. As urogenital, you can get glomerulonephritis. See, all of these are things that you will eventually see from people in the general public, or as if you're going in for nursing, you'll see these in your practice. Then you have group B strep. Group B strep, in the skin, you'll see in the wound infections, you'll see neonatal meningitis, neonatal sepsis, endocarditis, neonatal pneumonia. So this is something that can affect neonates quite a bit. Then you have enterococci, and in skin, you have wound or surgical infections, and then healthcare associated urinary tract infections. A lot of times we see enterococci, that's associated with urogenital. And healthcare associated urinary tract infections is because we introduce it into the bladder and into the urinary tract through catheters. Then the viridin streptococci. It can cause subacute endocarditis, but also it could cause dental caries. Viridin streptococci. Then you have Streptococcus pneumoniae, which actually in the nervous system can cause pneumococcal meningitis in adults. In the respiratory, you can actually have pneumonia, you can have otitis media, and you have sinusitis. And those typically are done by Streptococcus pneumoniae. Then you have Neisseria gonorrhoeae, and there you have the gonococcal ophthalmia, and usually ophthalmia is dealing with the eyes, so you have the infection in the eyes, and usually that's with during birth. And so the mother has gonorrhea, and then she transmits it to her baby, and the baby can have gonorrhea of the eye, basically. And then urogenital is gonorrhea, as we just mentioned. And so, with this one, with the you can have neonatorium. That's newborn. So it's newborn. So if you have gonococcal ophthalmia neonatorium, then you know that's from a baby got it on birth. Neisseria meningitis can cause meningococcal meningitis. Okay, so those are the two slides that I need you to know, and also I need you to know one more slide, which is 24. So it talks about MRSA. So 95% have penicillin methicillinase and are resistant to penicillin and ampicillin. That's when we talk about what they were talking about is they talking about the Staph aureus, and they say a few strains of Staph aureus are coagulase negative, but they say 95% of the Staph aureus have penicillin and are resistant to penicillin and ampicillin. And you can get methicillin resistant Staph aureus. These carry multiple resistance. These MRSA is very severe infection, and methicillin is one of the drugs that could actually take care of the Staph aureus for really bad infections until they came up with the methicillin resistance. So some strains have resistance to all major drug groups except for vancomycin. So methicillin resistant. Then now we have vancomycin that could potentially that can actually treat that. But then what ends up happening is we started seeing strains that were vancomycin resistant as well. But if you have abscesses, they have to be surgically perforated. That's where started talking about those biofilms previously, where you have so many and they wall themselves off and they create a barrier against uh, our immune system and attack. And so sometimes you have to remove abscesses surgically. And then systemic infections require intensive, lengthy therapy. If it gets really bad, it's very intense. So know this about MRSA with this methicillin-resistant Staph aureus. Know that's very resistant, but except for vancomycin. And then just going further, there's Staph aureus is resistance very strong, and then some of them are uh, resistant to methicillin, and then some are resistant to vancomycin. That's what we call VRSA. And then so we have new drugs that came out. That is the last drug of choice if you actually have a Staph aureus that is resistant to methicillin and vancomycin. All right, so those are the three uh, slides that I need you to know for the extra credit, 5 extra points. I think it's worth memorizing those and then study well. I'm not going to go through specifically for any other questions for chapter 18. I'll do those three, and I think you should do well. All right, you have a wonderful evening, and looking forward to seeing you soon.